So I just want to kind of go through the major groups of protists. And one of the things, so I'm going to send you guys a new schedule. One of the things I've been working on doing in this class this last year or so as I teach it is expand the conversation about microscopic life and contract some of the conversation about macroscopic life. And I know that maybe that isn't exactly how you'd like me to do it, but it also gives room, some more room for fungus and plants, which I had been kind of giving short shrift because I'm an animal centric person. And so I want to make sure that we like hit a couple of the highlights. There's no way that I'm going to go through all of the groups of protists. There's no way that you want me to ask you to remember features of all of the groups of protists. But I want to hit some of the highlights so you know some of the particularly important organisms and major like characteristics of some of the major groups. Even in the groups that we're going to talk about, there's a great deal of diversity. Protists are just these really diverse organisms. They have a lot of different ways of making their life. I think we're still probably working quite a lot on the taxonomic arrangement and how these organisms get grouped, and that might still change as we move forward in science. And so some of these groups are pretty diverse. So I'm not gonna talk about all of them that are in your book. I'm not gonna talk about all the subgroups that are in your book. I'm just gonna hit a few highlights. So this is the grouping. You can see that there are lots and lots of groups. Sorry, I'm gonna fix that. And so these are all of our major supergroups: the excavata, the chromovolata, the rosaria, the archoplastida, amoebozoa, and Ophisconta, I think is how you say it, but I don't remember. So these are our major groups. And then within those, you have some things that you might have heard of before, like diatoms or dinoflagellates or algae. Some of the things that we looked at in class like last week, like the euglena, red algae, land plants, right? Some other things that you have heard of are in these major groups. And then down here, we have our fungus and our animals along with the choanoflagellates. And we'll start on those when we start with fungus. Okay, this is another table. Again, I don't expect you to remember all this. I've just given you this for your reference. This is from a textbook that we don't use anymore, but it's a nice table. So I put it in there if you want to kind of like peruse the major groups and their subgroups and get some idea of the characteristics of these organisms. You can find that in that table. Okay, so the first group I want to talk a little bit about are the excavata. Their name comes from the shape of the cell itself. It looks like it has an excavated cavity in it, and that cavity is actually where some of the feeding happens in some of these organisms. These guys are interesting because they're anaerobes. That means that they live in a world without oxygen. And so they're going to not require oxygen for their energy acquisition, and in some cases are obligate to be in an environment without oxygen. Oxygen is actually toxic to them. And so that's sort of atypical for how we think about organisms. They also lack true mitochondria. They have these modified mitochondria or mitochondria-like structures, but they're not true mitochondria in the sense of how we think of mitochondria functioning. They're really diverse in their feeding. They can be predators, they can be photosynthesizers, they can be parasites. The two that I have here are both parasites that you might be familiar with. Giardia is a parasite that is pretty commonly picked up if you drink water, unfiltered water from a natural body of water. And so that parasite makes you pretty ill. You take some medicine and can get rid of it usually pretty easily. But that's a single-celled protist that causes that disease as opposed to a virus or a bacteria. Okay. The one subgroup that I want to mention in the excavates are the euglenozoans. These are the euglena and their relatives. These guys are all unicellular with flagella that they use for swimming. And they also all have ocular eye spots. So they have a pigmented eye spot that allows them to sense light and dark, usually, as opposed to making a clear image. They have some sense of light strength. These guys are mostly free living, although some of them function as parasites. The whole group has the same style of unique mitochondria. that is the thing that's used to characterize this group. And it includes things like kinetoplastids, which are things like trypanosoma, which is a individual protist that causes sleeping sickness, which is a tropical disease that you might have heard of before. So this is a really big group. The excavator is a supergroup. Within that, Euglenozoa is also a very big group. There are a lot of different types of organisms here, so these are just really broad characteristics. I'm giving you a couple of examples of things you've maybe heard of. I'm never going to ask you like which group is Giardia in. Okay, I feel like that's a scale of example that I'm not going to ask you. I'd rather you recognize that, most of the excavates are anaerobic as opposed to that Giardia is an excavate. Okay, these are just sort of for interest because you might have heard of some of these organisms. One of the excavates that we looked at last week in lab are the euglenids. These guys are photosynthetic or absorptive feeders, depending on the conditions. So these are one of those organisms that uses mixotrophy. If light is available, they will do photosynthesis. If light is not available, then they will bring food in and consume it absorptively. So you can see in this organism's body, they have the flagella that they use for swimming, the eye spot for detecting light. They have a contractile vacuole for osmoregulation, and then they have the chloroplasts dispersed in their body, and they tend to be green. Did most of you guys see these guys last week? Was one of them. They were super easy to find because they were really green and obvious under your microscope, and they were, yeah, kind of spinning around. Yeah, right. So most of these guys, when they're swimming in the microscope, you can't see how they're moving because their movement structure is doing the propulsion. So when these guys are moving around, their flagella are moving so quickly through the water that you're not able to actually focus in on it and see it. The same with the cilia. Some of you were maybe able to see the shadows of the cilia, but more often than actually seeing the individual cilia, you saw the water moving because they were creating a current by beating their cilia rather than actually seeing the cilia work themselves. Okay, the next group that I want to mention is the chroma alveolata, the alveolates. These are organisms that tend to be mostly photosynthetic and form an important component of phytoplankton. Can anybody tell me what phytoplankton is? Do you know what phytoplankton is? So these guys actually are plankton that glow in the water. And so the dinoflagellates, which is what's shown here, the blue right here is where a wave is breaking on the shore, and that is the glowing of the individual dinoflagellate bodies. And so these guys do actually bioluminesce, but they are part of a larger general group that has nothing to do with taxonomy, but just has to do with like role in the ecosystem called phytoplankton. And phytoplankton are the photosynthetic plankton. They are the producers of the open water. And so they're both found in freshwater and marine environments. And they basically float or feebly swim at the surface of the water. And they carry out photosynthesis and provide food for the animal-like organisms, the zooplankton, that feed on them. And collectively, the phytoplankton and the zooplankton are this like incredible nutrient source in the open water that's used by, everything from fish to baleen whales as their source of nutrition. So the dinoflagellates have two whip-like flagella that they use for swimming. They're predominantly photosynthetic. They can be either freshwater or marine, depending on what species group you're looking at. They're an important food organism for much aquatic life. Another one of the organisms that we hopefully saw last week in lab are the ciliates. This guy here is a paramecium of some sort, surrounded by many cilia that it uses for locomotion. You guys saw some little green ciliates and some pink ciliates last week. And then also the stentor that had the crown of whirling cilia was another one of the organisms in this group. These guys tend to have many different specialized organelles. Some of them shown in this organism are the contractile vacuole and the oral groove, which it uses for feeding. They also have an excretory pore called an anal pore that they use for eliminating waste that they sort of pull out of their cytoplasmic fluid and accumulate into this bladder-like structure and then excrete it instead of just moving it out through exocytosis. They are fast movers because of their cilia. They're known to respond to stimulus. So they'll respond to things like light and chemical signals in the water by swimming towards or away from those things. These guys also can be either freshwater or marine. Most of them are ingesters, and they use cilia to create a water current to bring food into their oral groove. They form a food vacuole and then consume that. But there are some ciliates that can be parasitic. And so not all of them are free living, but many of them are. The ones that we looked at were all free living predators. That's a great question. I have no idea whether ciliates or flagellates are faster, like in terms of like if you put them in a foot race, like rabbit in a hare over starting line, I don't know. The ciliates tend to be very zippy, and but then some of them, like the stentor, will actually temporarily anchor themselves, and so they're actually not moving hardly at all. It's probably also a body size thing, how much volume of fluid you're moving through and your shape. I would imagine the oblong ones are better swimmers than the big round ones, but I don't have a straightforward answer to that question. Um, it would be fun to test though. Like I wonder if we could like put a chemical signal and like race them towards it. Be fun to watch. Okay. Another member of this group are sometimes called the oomycetes or water molds. These are fungus-like protists. Remember we said protists could be sort of plant-like, animal-like, or fungus-like. The water molds are the fungus-like ones. And they have this distinctive hairy flagella. So they have a flagella with many little hair-like structures on it. And they are fungus-like absorptive feeders. So they are going to be absorbing their nutrients from the surrounding environment. They are decomposers of water, items in water like detritus and damp soil. They're found in damp environments, but some of them are actually found terrestrially. Um, they have long filamentous individuals that form colonies. And so this image up here at the top is not a single individual, but rather many, many individuals forming a colony around a food source that they're consuming, something that they're breaking down, floating on the surface of the water there. They're also the source of much plant disease, disease you might be familiar with in this area as a gardener, that we know we find in high humidity summers or in high humidity areas in our garden. So things like potato blight and downy mildew, and also sudden oak death. All of these are not actually true fungal diseases, but they're protist diseases of these fungus-like protists that participate in that. You may have seen these water molds. Also, if you've ever encountered like a fish that has died in the pond and it's decomposing, it gets this like fuzzy surface over it. That's often these water molds that are doing that feeding and decomposing. Okay. Some of you also probably looked at diatoms last week. They were in our brown algae group, and we had some marine diatoms. Most of the diatoms are marine, but there are some that are freshwater as well. These guys are all photosynthetic and tend to use pigments other than chlorophyll. And so they use pigments that are brown in color to our appearance and absorb different wavelengths of light than chlorophyll do. So they have a brownish appearance. These are actually under a special light microscope to help them to glow a little bit so you can see their body structures. And all of these shapes and the features and the sparkly bits of these shapes are produced by silica. And so silica is what we make glass out of. But their shells, their bodies are also made of silica. And so these guys contribute to sand. When they die, their little silica bodies become part of the sand that makes up the substrate of the surface and the base of the ocean. These also contribute to phytoplankton. I actually had the phytoplankton definition here: single-celled photosynthesizers that float in oceans or lakes. These are another food item for larger organisms or predatory organisms that are feeding in the open water. Okay. You guys have this? All right. Another example from the same group, the stamenopiles are brown algae. And I need a diver in this picture to give you perspective, because these individual kelp are tree-like in size. They're huge. A diver would just be like a little body up here in the water in amongst this forest of kelp. So that's not a magnified image. That's actually a very large macroscopic organism. These guys form multicellular aggregations, so they're not single-celled organisms like most of our protists are single-celled. These guys are multicellular. They are also part of what we call our brown algae. They have brown and yellowish pigments their appearance is brown or yellow to us. They absorb a different spectrum than chlorophyll. You also may be familiar with these if you guys have gone to the shore here in Massachusetts or in neighboring states. You find these brown algae with floats in them that sort of wash up on the shore, and they have these bubble-like structures in them. Those actually help to keep them buoyant in the water column. And so they're cellular air spaces. There's a structure with a big airspace in the center, like a bladder, and it functions as a float and helps to hold them up. So these guys tend to be multicellular. They have larger bodies. They're not microscopic. We're not looking at them under a microscope. This is, like my hand is the size of these individual filaments of algae. These are things you see on a much larger scale. Okay. I want to also briefly mention the group rhizarians, particularly the foraminiferans, which are a subgroup of the rhizarians. These guys are particularly interesting because they also contribute, their shells and their bodies contribute to our geological deposition. They have really thin thread-like pseudopods. So many of you found amoebas last week, and you saw the amoebas moving, and I said some of them were running because you could really see their cytoplasm streaming, but an amoeba running is kind of like this, right? So it's really fast. That projection, when the amoeba body sort of pushes out into a skinny projection, that's a pseudopod. Fake foot is what that word means. And they do that with their cytoskeleton. So they actually move cytoskeleton elements through the cytoplasm to create a structural element that is long and skinny. So rhizarians have these thin thread-like pseudopods, and they're not shown here because I have the skeletons shown as opposed to the living organism. But they use those pseudopods both for locomotion and feeding, much like amoeba do. So they can surround food with them, they can stretch them forward and use them to pull their bodies along. Unlike the dinoflagellates that had a silica shell, these guys have calcium carbonate shells. And so these are two examples of foraminiferans with their calcium carbonate shells. And when they die, their bodies actually settle to the surface, or settle from the surface to the bottom of the ocean, and they create limestone deposits. And so limestone is actually created by the bodies of these organisms settling out into layers over time. Okay, I think I only have a couple more. Amoebozoans, we looked at amoebas last week. The amoebozoans also use these pseudopods for locomotion. And so each one of these finger-like structures is a pseudopod made by streaming the cytoplasm out in one direction or the other. Amoebas can shape their bodies in all sorts of ways. They can kind of be long and skinny, or they can almost take on like a star-like shape where they've got lots of pseudopods out around their body. They are found both in freshwater environments like lakes and ponds. Most of them are predatory, kind of trapping other organisms and feeding them. Some of you were watching the stream of videos I had last week, and we were seeing amoebas sort of trapping little ciliate, um, paramecium, and other organisms. And they kind of like stretch their bodies around them and kind of get them into a space and then slowly close around them and pull them in through a food vacuole. Some of the amoebas are also parasites and can, sort of live in places like digestive tracts and fluid systems inside other organisms' bodies. And then one of the things that we're going to look at from this group this week in lab are the slime molds. And so I have some plasmodium. I'm culturing them again tonight. They will move towards a food item, so we'll hopefully be able to watch these guys move. And slime molds are pretty cool because they are individual amoeboid organisms, but they form these colonies and they kind of like ants can work together to form a big structure that is different than sort of like their individual body shape. And so these are single-celled organisms, but they form these really tightly connected colonies. And the individuals in the colony work like a single organism. So instead of each cell kind of being on its own, doing its own thing, the multicellular colony moves like a single organism and behaves like a single organism. They've been used in a lot of interesting studies. They respond to music. They respond to chemical signals, and so you can there's a bunch of interesting studies done with these guys where the individual colonies will actually react as if they're a single organism. And so this is sort of a colony growing. These runners tend to be searching for new foods. So the dark section is a mat where the colony is really quite big and there's a lot of individuals there. And then the things that look like veins or strings going out are tracks of individuals that are searching for food. And so there's some great, I'll send Jen the link, some time-lapse videos of them kind of moving towards a food source, and then you see the food source be completely covered by the colony, and then you see it move out and search for a new source. And so the organism, the organism is made up of all these individual cells, but they're actually behaving collectively as a group. And then the fruiting bodies are produced. I have an image here that'll show those. So when food becomes scarce, the cells aggregate into this big like slug-like mass, and they produce a spore-fruiting, spore-like structure that then produces individual spores and releases them, and each spore becomes a new individual, a new slime mold that can go off and participate in a new colony or start a new environment. And so when times get tough, all of the individuals actually kind of cluster together and leave the dark environment. They tend to like to live in the dark, and they move out towards the light. And they sort of sacrifice all of their cells to create these fruiting bodies and these spores for the next generation. And so those spores can resist desiccation and resist lack of food for a while. And then when they're in a good environment, the spores will split open and the new cells will carry on their way. So it's a way of kind of preserving your superorganism through a harsh time and kind of just make the next generation and package the next generation and save spores until environmental conditions improve and they can be released. So each individual needs to feed because they are individuals. They're not members of a multicellular body. But it wouldn't surprise me if there's some food transfer through the space between them. I don't know how actively they feed each other or how actively they share food. But the individuals who are immediately surrounding the food source are getting the best access, right? But the individuals who are at the next layer in the colony are not starving. And so either nutrients are being released from the food and are moving through through the space around them, they're able to pick them up, or there's some sharing going on. And I actually don't know for sure which of those two, how the mechanism works, which of those two is happening. Okay. And then the last group I want to mention are the red algae. We also saw some red algae. They were in the marine algae mix. So if you took something from the marine algae mix, you might have seen red or brown or green, but there was definitely red algae in there. These guys are also multicellular. They're photosynthetic seaweeds. They are found in the marine environment. Many of them are actually found in deep water and are able to absorb some of the light that trickles down through the water column. Their red pigments are what we see, so they take on a pinkish or red color. They also accumulate and deposit calcium carbonate. And so when these algae are growing on reefs, they deposit additional calcium carbonate onto the reef and help to build the reef structure. How many of you have ever been to a reef area? A couple of you. Have you ever been snorkeling in an area that has calcareous algae or coralline algae? Heard that term before. These guys are what makes up the coralline or calcareous algae. And so it has a different shape and structure than coral, but it is also hard. And that's because they're able to accumulate calcium carbonate in their bodies and deposit it, and they create these rigid structures. And so they can, if they're on a reef, they'll contribute to reef building. If they're not on a reef, they can actually form areas where there's like a hard, robust structure from their bodies forming um, on rocks or on the open surface. They're also harvested and used as a source of carrageenan, which is a thickening agent or a food stabilizer. You'll see it listed in the list of ingredients. You don't know what they are at the end of many food items. It actually comes from the bodies of these algae. And so they're harvested to create that particular food additive. Okay, I think, yeah, just have one more. So the last group, I thought red algae were the last group. The for real last group are the green algae. Um, green algae are the algae we're most familiar with, the ones we see most often in our environment. They're ubiquitous in freshwater and saltwater environments alike. Many of them are unicellular and form colonies, but other individuals are actually multicellular. So many of you probably saw this Spirogyra last week. I would have pointed it out to you if you asked me for help with green algae, because this is my favorite green algae. It's just really pretty. Each one of these boxes is a cell, so each rectangle is a cell, but then the filament that you see is actually a colony of many cells functioning together. These guys are all photosynthetic, predominantly freshwater, but again, there's some marine ones. They may form these interdependent colonies where individuals actually function in a multicellular way within the colony. So some of them give up some of their needs, or some of their may lose some of their photosynthetic ability in order to do something else, like fix nitrogen or contribute to the colony in a way. Although they remain individual cells, they can function in this more interdependent manner. So they're kind of that link between being a colony and becoming a multicellular organism when you start to see that kind of behavior. Some of them have been tested as biofuels with varying success depending on the species. Some of them are actually pretty good biofuels if you have the capacity to grow enough of them. And these are our ancestral plants. So this is the link between our protist world, our mostly single-celled protist world, and the land plants. And so there's a green algae known as chara, which is probably the link to the ancestor to the plants that moved on to land and developed their land characteristics. So they share the same pigments as our green land plants and have a lot of commonalities with them, but they're all still reliant on being in an aquatic environment. Okay, so it's a really broad stroke overview of protists. I am not a protist expert. Obviously, you guys asked me questions I couldn't answer. I don't have a deep dive on these guys. Certainly don't expect you to. I mostly want you to know the breadth of diversity. Like these are all of the different kinds of things that are in this massive group called protists. And they have a lot of different ways of making their living, and they contribute incredibly to our ecosystems as major food sources for other organisms in the food web, as photosynthesizers, as limestone, as silica, right? They have as reefs. They play a major role, even though we don't see them individually or necessarily think about um, what the organism is. If we see a clump of algae, not really thinking about how many individuals that is in a colony creating that. Okay. Does anybody have any questions about these guys?